The next topic which we are going to start is protein in lens. So what are all the proteins which are associated in the lens? These are water soluble and water insoluble proteins. Okay, water soluble proteins and water insoluble proteins. Water soluble proteins are also known as crystallines. Okay, and these proteins are in a form of crystals. Okay, as the name suggests, crystalline proteins. So in a crystal form. Now these crystalline proteins are further of three subtypes: alpha, beta, and delta. The major we are going to study about the alpha and the beta. The alpha crystalline is the largest crystalline. The beta crystalline is the major portion forming crystalline. Approximately 55% of the water soluble crystalline is formed by the beta crystalline. Okay, now the alpha crystalline is a heat shock protein, very important over here, is a heat shock protein, and it prevents denaturation of the crystalline. Okay, so alpha crystalline, a water soluble crystalline, this is a heat shock protein and it prevents denaturation of other crystallines. Now coming to the water insoluble proteins, these are urea soluble or urea insoluble proteins. Okay, so urea soluble water insoluble proteins are cytoskeletal proteins. Okay, now these proteins, these are distributions leading to the cataract formation. Okay, so the cataract formation in the eye is due to urea soluble water insoluble proteins. Okay, now the urea insoluble proteins, they help to maintain the transparency of the lens. Very important. Urea insoluble proteins or water insoluble proteins, these helps to maintain the transparency of the lens. For example, MIP26 or aquaporin in O. Okay, these are the examples. What are the examples of urea soluble water insoluble proteins? These are vermentin, flensin, and faculin. Okay, these are the three examples. Now what all are the water soluble and water insoluble proteins? The water soluble proteins include HM1, HM2. The water insoluble proteins include HM3 and HM4. So water soluble includes 1 and 2. Water insoluble includes 3 and 4. Okay, now the water insoluble proteins, there is an increase in concentration in cataracts. Okay, so in cataract, what is going to increase? Water insoluble proteins are going to increase as we started. Okay, now the HM4 alone shows increased concentration in nuclear cataract. So in a particular cataract such as nuclear cataract, HM4 plays a role, very important role of HM4 in nuclear cataract. Okay, so if HM4 increases, then now some antioxidants in the lens. Okay, so what are these antioxidant which you can see in the lens? These are superoxide dismutase, the first one. Okay, then comes your catalase, then glutathione reductase, vitamin C and E, very important. Remember, the vitamins are C and E for antioxidant purpose in the lens, not A. Now what other things are antioxidant over here? So due to low oxygen level around the lens, okay, this can lead to also antioxidation, okay, in the lens. Now in vitrectomy, okay, so the vitreous tumor is being removed over in this surgery, vitrectomy. So there is a low oxygen level in is lost. So in vitrectomy the oxygen level is lost. Okay, this leads to increased incidences of cataract. Okay, so vitrectomy, oxygen level lost. This leads to increased incidences of cataract. Okay, now what you have to remember is vitamin C and E are have important role in the antioxidation. Vitamin A doesn't have any role. Okay, vitamin C and E have. Now, what is lens paradox? Okay, so lens paradox, a very famous question, and the lens paradox, it is seen in old ages, old individuals. So what basically it's lens paradox and how it is occurring. So it is a balance mechanism. First of all, lens paradox is a balance mechanism in old ages. So there is a increased curvature of the lens and decreased refractive index. So what do we mean, or what will happen by this increased curvature and decreased refractive index? So basically when your curvature of the lens increases, the power of the lens increases, and the refractive index when decreases, the power of the lens decreases. So these both mechanisms are occurring simultaneously, and they lead to balance of the vision in the old individual. Okay, so in old ages, this is how your lenses are going to be working to balance your vision, okay, by lens paradox. Very simple. Now the next very important topic, metabolism in lens. So in metabolism in lens, there is glucose diffusion incidences into the aqueous humor mainly, and in the vitreous humor also. So basically the glucose is diffusing inside the aqueous humor and the vitreous humor, and more it is diffusing in the aqueous humor. So mainly in the aqueous humor. Now the glucose is further metabolized. Okay, the glucose is further metabolized by anaerobic glycolysis, and 80%, more than 80% of the glucose is metabolized in the lens by aerobic glycolysis. Now another thing comes over here is Krebs cycle and the HMP shunt. So approximately the glucose is metabolized by Krebs cycle and HMP pump, or you can say it. So it is metabolized approximately 15%, okay, by Krebs cycle or HMP shunt. Now the very important aspect of metabolism of glucose inside the lens, that is sorbitol pathway. Less than 5% of the glucose is metabolized inside the lens by the sorbitol pathway. Now let us discuss about the sorbitol pathway, why it is playing role in metabolism. So sorbitol is not itself playing a role, but after activation it plays a role. Like the sorbitol pathway, when it is activated in diabetes, in sugar cataracts. Okay, so in diabetes patient there is lots of sugar, or glucose I should say rather, there is lots of glucose to metabolize, and so we will metabolize that glucose inside the lens by sorbitol pathway and anaerobic glycolysis. So that leads to sugar cataract. The next thing about sorbitol pathway activation, it could be activated by galactosamine. Okay, so galactosemia can lead to also activation of sorbitol pathway. Now let us see the complete mechanism from the glucose metabolism to sorbitol formation, and ultimately which would lead to cataract. Okay, so the glucose by aldose reductase, as we discussed, it is converted into sorbitol. Okay, so one sugar is converted into another type of sugar basically. Now this is controlled diffuse out of the lens. Okay, so sorbitol, this control diffuse out of the lens, and it accumulates inside the lens. That is why sorbitol accumulates inside the lens. As the sorbitol gets accumulated inside the lens, it increases the osmotic pressure inside the lens. As it increases the osmotic pressure inside the lens, the fluid is drawn inside the lens. Okay, so the fluid is drawn inside the lens. Then this leads to opacity. Then this is called as cataract. So what basically is happening? Sorbitol cannot diffuse out of the lens. It accumulates in the lens, increases the osmotic pressure of the lens, which leads to fluid which is inside the lens. It increases and this leads to opacity, which ultimately causes cataract. Okay, very simple. Now there is a active process. We can transport the ascorbic acid in the lens by a active process by sodium dependent SVCT2, and this is a active process of transporting of ascorbic acid into the lens. So that was all about our topic. The next we are going to study is lens acquired cataracts.